Okay, DNA structure is a lot easier than protein structure, and so hopefully you'll have an easier time understanding it. In the early 1900s, we knew that chromosomes, the DNA were found in chromosomes, and we know there's some proteins in chromosomes as well, by the way. And chromosomes consisted of DNA, deoxyribonucleic acid, and these proteins called histones. We also knew that it was the heredity, that these chromosomes were hereditary, had hereditary information. But the question was it the DNA or the protein that was the carrier of that information? And as we discussed in class, if you think about it, and by the way, I would expect a question on it in the exam. Which of the two would you expect would have the information, the genetic information that would describe such a complex organism as a human being? The molecule that had only 20 variables, it only had 4 variables, DNA, A, G, T, or C. Or would you expect that it would be the molecule that had 20 variables, the 20 amino acids found in proteins in your body? Well, I hope you said it would be, you'd expect that protein would be the information carrier, the genetic information carrier. I think every scientist at the time, or almost every scientist at the time, was convinced that it was protein that carried the genetic information. As it turned out, the genetic information was carried in DNA. We know that today. Let's look at what other information we know about DNA. The chemical structure of DNA, we know, has 3 groups, and the 3 groups are focused on the phosphate, and you've studied phosphate as a functional group. It will be tested on the exam. You have the deoxyribose, and remember, please remember that deoxyribose, the whole difference between deoxyribose and, let's say, and ribonucleic acid, is that this second position here does not have an oxygen, no oxygen in DNA. In RNA, there is an oxygen there. It's an OH. So don't get that confused. The other thing to remember is that there's a base here. And in DNA, we have adenine as a possible base. We have guanine as a possible base. We have cytosine as a possible base. And we have thymine as a possible base. Those are the 4 nucleotides that we can, that we will find in DNA. Now, thymine is an interesting molecule in that when we make RNA, this molecule, this doesn't fit, this base doesn't fit. And so it's replaced in RNA by uracil. So let's try to remember that in DNA is found in thymine. So if you see a molecule that has a T and no oxygen here on the second carbon, and there's a T in it, then you know it's deoxyribonucleic acid. It's DNA. If you find a U in it, or you find an oxygen in the ribose sugar, then it is RNA. So this is a nitrogenous base, and we'll look at those nitrogenous bases next. These are 4 possible nitrogenous bases that could be here. So in proteins, we had 20 possible amino acids, and we had that backbone of NCC, and then you stuck any one of those 20 amino acids on the side. In DNA, there's only 4 possible variables that fit in the first position of the sugar. This is the sugar. It's a ribose sugar, all right? And phosphate is sticking out as part of this molecule as well. The phosphate and the sugar, they make the backbone. They don't change. So as you make a double helix, and we'll look at that in a minute, as you make a double helix in your body, the backbone of that double helix doesn't change. It's sugar, phosphate, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down the molecule, billions of nucleotides long. The guanine changes. It's analogous to the R group in proteins, and that base can be any one of these four, and which one of these four is important. We'll look at that. But this sugar and this phosphate are the backbone, while this is the R group. So here, again, I'm going to say this is the monomer of DNA, right? Where DNA itself is the polymer. So the monomer of DNA is a nucleotide. Here in this slide, it's printed very clearly for you the three components of DNA. It's a nitrogenous base. We'll look at why it's called nitrogenous in a minute. And there's four bases, A, T, C, or G. These are the possibilities in DNA. Remember, this T turns into a U because T doesn't fit. So you need a U in RNA. Let's take a look next. Here you have two groups of nucleotides. You have the nucleotides that have a double ring structure. Here's one ring here, and here's another ring. So double ring structure. Remember, one of the properties of carbon, which again will be tested, is they can form rings. You know, it's not just straight chains. And so these rings, double ring structure, we call them purines. And there's two of them. There's an A and there's a G. You'll notice that the difference between the A and the G is you have a carbonyl group here, and then the amine group gets moved down to here. But the bottom line is that you have a double ring structure, and there's slight difference between them, and one's adenine, the other's guanine. And these are known as purines. In pyrimidines, you have pyrimidine has one single ring. Notice that obviously the difference, I hope you see it. This is two rings, this is one ring. All three of these have only one ring as its base. The base only has one ring. Now you'll notice also that the phosphate is here, PO4, right? And the sugar is here, the ribose, the deoxyribose. Notice the second carbon, there's no sugar, there's no oxygen. There's no oxygen on any of these because this is DNA. This is a mistake by the printer, I would assume, that since this is only found in RNA, you would find this would never be an H group. This would be an OH, right? Because RNA would have an O there. So this is a mistake by the maker of this illustration. So this would be an OH here, and that's because this is ribose, and these four are deoxyribose. There's no oxygen on the second carbon. The first carbon has a base attached. Here is a better drawing than the previous slide because the oxygen is shown in its proper position, and the phosphate's here. And this phosphate, sugar, phosphate, sugar, phosphate, sugar, this is how they're bonded to each other. The one is bonded here, then this phosphate would be bonded to this. There's an OH here in the third position, so this OH can leave and a hydrogen from, so we can have dehydration synthesis, and then we can form an ester bond between one of these oxygens and this third carbon. So it's pretty straightforward. Now, Watson and Crick developed a model, and we talked about these two gentlemen. Well, I'm not going to call them gentlemen. These two guys, Watson specifically. We talked about what a... I don't want to say anything negative, any more negative. I don't want to go down to his level, but you know what a disagreeable individual this person is. He probably, in my opinion, is an embarrassment to biologists, and his viewpoints are disgusting at best. But he and Crick did develop a model of the DNA double helix. He did steal that information. We'll talk a little bit about it again on the next slide, and it's something that is again a shame in the history of biology that when people act this way. And it's something maybe you can take a lesson from if you act this way, and even if you become famous and you might be powerful for a short bit of time, that black mark will never leave you. to this day we talk about it, and Watson is an old man, and we'll be talking about it probably for the next 100 years, the story of how Watson stole the information to come up with the development or the structure of DNA. So DNA consists of 2 chains of nucleotides, and these 2 chains, one on one side and the other, this is 2 separate molecules. So when we say 2 chains, I hope you think when you see 2 chains, you also think 2 polymers, right? So chains, remember, polymers. So we have these 2 polymers, 2 molecules, 2 macromolecules of deoxyribonucleic acid. When they come together, they match up and they form this double helix. So again, Watson-Crick, they used data that Wilkins and Roslin came up with. And I believe in class I said it was Wilkins that helped steal that information. It was not Wilkins, it was somebody else in the same lab. And that individual took this picture and stole it, copied it, showed it to Watson and Crick. They were working on a DNA molecule themselves. And they freely admitted that this happened. In fact, when they published, they freely admitted it. And Watson and Crick stole that information, and that took this woman hundreds of hours of her life to come to this picture of B DNA, and there's another picture of A DNA as well. And her work, probably her work alone, and some of the people and more than one person that was her contemporary and her superior even came out and said clearly that her work was phenomenal and was the basis of Watson and Crick's deciphering of the structure of DNA. If it wasn't for this picture, Watson and Crick probably would not have gotten that structure of DNA the way they did. And this stealing of this picture is what changed the history of DNA and who got credit for it. So Rosalind Franklin died in their mid-30s of ovarian cancer because of her work with radiation to try to get this picture. When she left the lab, a couple of years after Watson and Crick published their work in Nature. She left the lab and went to go work on tobacco mosaic virus because she wasn't allowed to work on DNA. That was the agreement between her and the person who ran the lab she worked in, that even though she came up with the picture that made everything happen, these 2 people stole that picture along with a third conspirator. That person that ran that lab said that she made an agreement with this person never to work on DNA again. Who knows what kind of amazing work could have came from her had she been allowed to do what she could. And she went on to work on tobacco mosaic virus and ended up doing some really great foundational work on the structure of viruses. So if anyone should be remembered as a great scientist, it's Rosalind Franklin. Everything she did was about pure science, and her work drove a lot of what we do today. These 2 guys, like I said, although they stole this, they did figure some things out, and I just hate to say, I will never, I think in the rest of my career, will I ever be able to describe their work as clearly as Watson and Crick did this. Watson and Crick did not do this. Rosalind Franklin did it, Watson and Crick stole it and worked on it. X-ray pattern suggests the structure of DNA helix, and again, Watson and Crick did figure it out and published it in Nature. Part of the work that they did, Watson, Crick, Rosalind Franklin, everyone who was working on DNA, it was based on this information that Chargaff got from just a simple, maybe not so simple, but some work in biochemistry where he looked at a bunch of different samples from DNA, and he found that every time he looked at DNA, no matter where it came from, no matter how much of any individual nucleotide you had the same ratio of percentages. So if you had 50% A's, you had 50% T's. If you had 30% G's, you had 30% C's. The G's and C's and A's and T's always equaled in the percentage of how much there was. So this led people to the conclusion that something must be happening. There must be some kind of bonding happening between A and T when you make DNA, which of course allowed Watson and Crick to figure out that there were two molecules, and the two molecules were bonding. The A's in one molecule was bonding to T's on the other, and the C's in one to G on the other. Not covalently bonding. Again, I'll say it again very clearly, not covalently bonding, but hydrogen bonding. Here are these two people describing their structure that they came up with. There's Watson very arrogantly describing it. What's key is that the ladder itself is made of that phosphate sugar backbone, but the rungs, and again, rungs are the steps or the little dowels in a ladder that you're able to grab onto and walk up. A lot of people don't know what rungs mean, but the bases are the rungs of the ladder. And the rungs, again, it's hydrogen bonds that hold those rungs together. A is paired with T and G with C. Here is the backbone. This red section here is our nucleotide. There's the phosphate, the sugar, and the base. Phosphate, sugar, base, phosphate, sugar, base. Each of these, there's 3 nucleotides here and 3 nucleotides here. This is one molecule. There's one molecule here on one side. And there's another molecule over here on the other side. So this is one molecule and this is one molecule, and they're hydrogen bonded to each other. These are hydrogen bonds. Okay, these are not covalent bonds. This is not a double bond and triple bond. These are 2 hydrogen bonds, 3 hydrogen bonds, 3 hydrogen bonds. Between G and C, there's 3 hydrogen bonds. Between A and T, there's 2. This becomes important later when you're trying to calculate how many G's and C's there are in anything. You can actually calculate that depending on how much heat it takes to separate them. So it's interesting. And remember, and I know this was a confusing point, but whenever you have hydrogen bonds in, the more hydrogen bonds you have, the more strongly they're pulled together. So this is phosphate, this is sugar, there's the base, this is the backbone. You notice that the backbone doesn't change on either side. The backbone is the backbone. A's are bonded with T's and G's with C's. 3 hydrogen bonds for G and C and 2 for A and T. You'll notice also that these are parallel, meaning that obviously, or at least I hope it's obvious, that one is next to the other. But they're flipped. You'll notice that with one molecule, you have the 5' on one end, and you'll find the 5' on the opposite end of the other molecule. So because they're flipped, we call them anti. All right, so they're anti-parallel, and that's important because you'll hear that term anti-parallel again. So they're flipped, and that actually makes it a little more difficult when they're making copies, when the enzyme is making copies of these 2 strands and making sure that everything is in its appropriate place. So here you have, again, the sugar phosphate backbone. Here you have a sugar phosphate backbone. It does not change. Here you have your bases. This is one molecule on this side. This is another molecule on this side. Together, when they come together, the A's form 2 hydrogen bonds between the A's and T's, and the G's and C's form 3 hydrogen bonds. And this is called base pairing. This is base pairing. So make sure you know that this idea of hydrogen bonding is indeed base pairing. And of course that the nucleotide is a phosphate sugar and base. When this base pairing occurs, because of all the hydrogen bonding, the tension, the geometry of it, this turns out to be a double helix, meaning that two different molecules form this helical shape. Let's be clear on this. See if I can't be absolutely clear on what we're talking about here. When you're talking about the backbone versus the rungs of the ladder, or the bases. So when we're talking about the backbone, you're talking about the part that doesn't change, the phosphate sugar, and that's here. And there's two of them, right? Because there's two molecules of DNA here. And in the center of this molecule is this interesting, these bases, and they're hydrogen bonding with one another. That you'll find here, right? That this part here is this, and when you're talking about the sugar phosphate backbone, you're talking about this part of the DNA molecule. So the rungs, which are these kind of dowels, or the things you grab onto, or the steps of the ladder, they're in here. These are the rungs, and that's these bases hydrogen bonding with each other. That's what that line represents. Remember, these bases are pretty complex. They're made of a lot of nitrogen. That's what you saw that in the previous slide. If you didn't rewind it and look at it in a couple slides back, look at what the nitrogenous bases look like, and you'll see there's a lot of nitrogen in them. And that makes sense. If you're going to have a lot of hydrogen bonding, you need either fluorine, oxygen, or nitrogen connected to hydrogen, and so you have that with the bases. I decided to go ahead and let you look at this slide again on purpose, and focusing on that whole nitrogenous base. You see here there's a base, and as we said before, this is a purine because it has two rings, and this is a pyrimidine because it has one. But look at how much nitrogen is in it. There's a lot of nitrogen in these bases, and that's why we call them nitrogenous bases. There's that sugar phosphate backbone. This would be connected to this, which would be connected to this. So the dehydration synthesis is happening between these. As we talked about with proteins, dehydration synthesis happens within the NCC backbone. The R groups in proteins don't have anything to do with the breakdown or buildup of proteins. It's the backbone that has to do with that. And it's the same with DNA. It's this backbone that's connected one to the other. These bases stick out and interact with one another. And so guanine hydrogen bonds to cytosine. And so this orients itself so that it gets three hydrogen bonds between these this molecule, this base, and this base, and A and T come together in DNA to orient itself again to get two hydrogen bonds. And remembering that this should be OH because it's found only in RNA, and RNA should have a OH. But the same here when you're talking about putting uracil with adenine, this orients itself so that this base is, remember this is on one molecule, this is another. This base aligns itself to be able to have two hydrogen bonds between it and it. There are, I believe I sent you two better DNA animations, and if not, I will send it to you soon. Use those. These are the two DNA animations. This one doesn't work, I don't think. Yeah, this one doesn't work, and this one is not too exciting. The other two are that I sent you are better. That's it for DNA animation, DNA structure. That's really all you need to know if you can get that in your head. And we've gone over it in class. We went over the same PowerPoint in class, I think in all three classes. We finished the DNA structure. If we didn't, here it is. And we certainly have discussed the highlights and the important points that are going to be on the exam. All right. Remember, this is just the beginning. Next, we have to figure out how do we make more DNA? How does DNA mutate? How does DNA make RNA? How does RNA make protein? And so that all is next on what's going on with this molecular machine. All right, have a good day.